So, girls, today I'm going to speak on this cloning vectors. I had already mentioned so many times, right, when we did on gene cloning, and also I had explained on TI plasmid, which is one of the vectors used for gene delivery, right? So I'll just go fast, explain. I'll just give an introduction to these vectors, a brief discussion on these vectors, and also on TI plasmid and lambda phage, which is these two, they are mentioned in your syllabus, so we have to study in detail about TI plasmid and lambda phage. So coming to the cloning vectors, in molecular cloning we have studied that the vector is a DNA molecule used as a vehicle, right? It is used as a vehicle to carry the foreign genetic material into another cell. You have seen that in cloning, where a gene of interest, a DNA molecule, is inserted into a vector, and this vector, it acts as a vehicle carrying that gene of interest into the host cell, the E. coli cell, where it can be replicated and it can be expressed. A vector containing a foreign DNA is termed as a recombinant DNA. This also I had explained in detail that recombinant DNA. Why? Because recombinant means recombine, the recombination of two DNA. Why? Because the vector itself is generally a DNA sequence, right? And it consists of an insert. Insert meaning our gene of interest. So when two DNA, they recombine, that is why this whole process is termed as the whole process is termed as a recombinant DNA technology, and this DNA molecule, that is the vector with the insert, is termed as the recombinant DNA. And a larger sequence, and you have seen that in a vector we have a portion where where we can insert our DNA of our gene of interest, and it also has a larger sequence that serves as the backbone of the vector. And we have seen that in the case of plasmids, right, those circular DNA molecule which acts as vehicle. So the four major types of vectors, they are plasmids, viral vectors, cosmids, and artificial chromosomes. Okay, and of these four major types, the most commonly used vectors, they are plasmids. So the plasmids, viral vectors, and cosmids, most of these, they are the natural vectors, okay, whereas artificial chromosomes, they are the plasmids which are artificially synthesized. And if you take a look at the whole, okay, we will come to the characteristic features later on, but if what is common to all the engineered vectors, the features, the characteristic which is common to all is an origin of replication. Okay, the presence of an origin of replication, and it should contain a multi-cloning site and a selectable marker. Origin of replication, for replication to occur, right, to begin. And a multi-cloning site, that is a site um, where we can cut with the restriction enzymes and we can insert our gene of interest. So why multi? So that you can use multiple restriction enzymes to cut at different sites. Because I had mentioned that, for example, like EcoR1 and BAMH1, those are the two different enzymes, restriction enzymes, and they will cut in different sites. So a vector, it must have a different sites for those two different restriction site. So it should have a multi-cloning site where we can insert different types of our gene of interest, and also it should have a selectable marker. A selectable marker because when you study gene cloning, you remember that how we have to screen whether the vector they contain our gene of interest or not, right? Whether the bacteria has taken up the vectors inside those which has our gene of interest. So to know that these selectable markers, for example, those which has the antibiotic resistance. Now, if you grow these microorganisms, the host cell in the meat in a plate, those which has the resistance to the antibiotic, they will grow, and those which does not have the resistance, they will die, they will not grow. So all the vectors, they should have this selectable marker. And the purpose of all the vectors altogether, any vector, is that it transfers the genetic information to another cell. Okay, and the purpose of a vector is typically to isolate and to multiply, to produce multiple copies, or express the insert, meaning the DNA, our gene of interest, inside the host cell. So all the vectors, they may be used for cloning and are therefore referred to as the cloning vectors. But the thing is that there are vectors which are designed specifically for cloning, while others may be designed specifically for other purpose, okay? Not for cloning, but for other purpose such as transcription and protein expression. And these vectors, which are designed specifically for expression of transgene in the target cell, they are called expression vectors. Why? Because you have seen in gene cloning that when you insert the DNA and it produces multiple copies, and it does not transcribe or translate, it just produce a clone, a multiple copy. So those are the cloning vectors. But some vectors, they are able to transcribe and translate, and they are able to produce protein. So since they are able to express, those are referred to as the expression vectors. And such vectors, they generally have a promoter sequence. You know that a promoter sequence is needed for the transcription process, right? So these, they generally have a promoter sequence that drive the expression of the transgene or insert. So we have seen that the expression vectors, they are actually different from the cloning vectors. But the simpler vectors, called the transcription vectors, they are only capable of being transcribed, okay, but not translated. So that is why they are referred to as transcription vectors. However, they can be replicated in the target cell, but they cannot be expressed. Unlike the expression vectors, and expression vectors meaning they are able to express themselves. These are used for producing proteins. Okay, that means they are able to produce to uh, carry out translation. But transcription vectors, they are used only to amplify their insert. Okay, so next I just want to mention on the characteristic important features of the cloning vector. I had already mentioned few, like some of the characteristic features of cloning vectors. They should possess an origin of replication so that it can self-replicate inside the host cell, and also it should have a restriction site so that the restriction enzymes can cut and insert the target DNA. And it should have a selectable marker with an antibiotic resistance gene, okay, that facilitates screening of the recombinant organism. Like I had given example, suppose if they have this selectable marker with an antibiotic resistance, So how this will help in screening those those E. coli cells which has taken up a recombinant DNA inside the vectors, okay, which has taken up those vectors inside their cell, they are able to resist, okay, because they have the selectable marker. So they are able to resist the antibiotics and they will grow. Whereas those which did not take up the vector inside their body, those bacterial cell, they will die. Okay, because they will not be able to resist the antibiotic. So that is just an example. So that is why a selectable marker is uh, important, because it will allow the host to grow on a selective media and it can selectively amplify the specific vector in the host cell. Next, it should be small in size. Okay, it should be small in size so that it can be easily integrated into the host genome, into the host cell, and it should be capable of inserting a large segment of DNA. But uh, like for example, like plasmids, they are not capable of inserting a very large segment of DNA. So that is why we have many other forms of vectors. And a good vector, it should have a possible multi-cloning sites, multiple cloning sites. Okay, as you can see here in this figure, eco r1, then cla1, then hin3, then eco rv, all these they can cut at different sites. And those sites where these restriction enzymes can cut, the DNA insert can be inserted into this position, into this region. So these are the multiple cloning sites, and it should be capable of working under the prokaryotic as well as eukaryotic system. It should be easily isolated from the host. Also, just remember that important point. It should be easily isolated from the host. So you see, these are the important features I have given in the notes. Also, you can study from the notes also those important features of a cloning vector. So, coming to the types of the cloning vectors, they are of the following types. Okay, so I'll just give an introduction to these vectors, as you don't need to study in detail, but we need to study only on the TI plasmid and the lambda phage vector. So, the first type of vector, they are the plasmid vectors. These plasmid vectors, they were the first vectors which are used in gene cloning. Okay, and they are usually found in bacteria, eukaryotes, and archaea, and they are actually the natural vectors. Okay, they are the natural extra chromosomal self-replicating DNA molecules. I had already always told this before also, that these plasmids, they can be self-replicating and they are actually the DNA molecules, okay, extra chromosomal DNA molecules present in the bacteria, and they encode proteins which are necessary for their own replication. So coming to the second type of vectors, the bacteriophage vectors. Just from the name, bacterio comes from the term bacteria and phage meaning virus. So these are also, they are actually types of vectors present in a phage, okay, in a virus. They are actually viruses which acts as a vector. Now, one of the example which we are going to study in detail, the lambda phage vector is actually a bacteriophage vector, and a TI plasmid is a type of plasmid vectors. Next, coming to the cosmids, they are actually the hybrids. Okay, they are the hybrids between the plasmids and the bacteriophage vectors. They are actually the hybrids between these two, the first two vectors, and they are artificially synthesized. Phage mids, even the phage mid vectors, they are artificial vectors, they are not natural. So just remember that the natural vectors here, they are only the plasmid vectors and the bacteriophage vectors, whereas the rest, they are the artificially made vectors. Okay, they are artificially synthesized. And one more vectors here they did not mention, the human artificial chromosomes. Bax and yaks, they are the bacterial artificial chromosome and yeast artificial chromosome. So all these other vectors I have given in the notes, you can just go through. So coming to the TI plasmid, I had already explained last time. So I will just remind you that this plasmid is a plasmid used to obtain from the bacterium Agrobacterium tumefaciens. This is a pathogenic bacteria, a gram-negative soil bacterium, which is a plant pathogen. Remember, girls, that it causes crown gall disease in many dicotyledonous fruits and other vegetables, okay, other plants. So this it causes a tumor. This ti plasmid, why the name ti? It's for tumor inducing. So the full form of ti is tumor inducing plasmid of Agrobacterium tumefaciens. It has been developed as a vehicle for introducing foreign genes into the plants. Why and how that is possible? So let us take a look at the plasmid structure first. Now you see the presence of origin of replication, then the Vir genes, and the tDNA region, sorry, and the opine catabolism. Remember, in the test also I had given one question on which part of this TI plasmid would be transferred into the plant. It is the tDNA region. Okay, so that means when the plant is wounded, it produces some chemicals. Then these chemicals, they activate the Vir genes. Then these Vir genes, they will facilitate the transfer of tDNA region inside the plant genome. And due to the presence of this gene for auxin and cytokinin as well as opine, what are opine? They are the specialized amino acids which acts as the source of nutrients for bacteria, right? So what happened when it's like this TI plasmid, you can see here, it acts as a vehicle. This TI plasmid, it is present in Agrobacterium tumefaciens, right, inside the bacteria. Now, when these bacteria, they infect the wounded plant, they will transfer this tDNA region with the help of the TI plasmid, which acts as a vehicle for transferring the tDNA from the bacteria inside the plant. And when the opines, they are specialized amino acids, they are synthesized in the plants, but then they will be catalyzed in the bacteria itself, because bacteria, it has this opine catabolism gene in the TI plasmid. You can see here the opine catabolism is for catabolizing these opines which are synthesized in the tDNA region. So these are the main region, okay, the auxin and cytokinin genes which induces cell division and proliferation. That is why leading to the crown gall disease, okay, tumor formation. And the opine gene, they will synthesize the opines like octopine, nopaline, and one more, the agropine. So these TI plasmid, they are actually categorized into different categories. They are classified according to the opines they produce. Okay, some they produce nopaline, so they are referred to as nopaline plasmid. Those which produce octopine, they are referred to as octopine plasmids. And this tDNA region, as you can see, it is flanked by a 24 base pair direct repeat sequences called border sequences. You can see the left border and the right border. So after observing the how the bacterium it uses this TI plasmid for transferring the tDNA from a bacteria to the plant. Thus, the scientists they can use this particular TI plasmid of this bacteria as a they can use it as a vehicle. They will cut this tDNA region. Okay, some portion of this tDNA region will be removed, and we can insert our gene of interest, our DNA of interest into this portion. And when the agrobacterium tumefaciens it inserted this TI plasmid into the plant, then when it replicate our gene of interest, our DNA of interest also will get replicated, because we see here the presence of the origin of replication in the TI plasmid. So this is the overall structure. And the Vir genes, as I had mentioned already, that they are of 8 different types, Vir A to Vir H. Okay, and their different functions I've given in the notes. You please go through. They have different functions, but they work together, and their main function as a whole is to help facilitate the transfer of the tDNA. Just remember, they help in the successful transfer of tDNA into the plant genome. And you know that the opine catabolism region, it functions in the uptake and metabolism of opines produced in the tDNA region. And we have the ORI, that means the origin of replication, responsible for DNA replication. So those are the structures of the TI plasmid, okay, the different regions in the TI plasmid. And so that is why the TI plasmid it is used as a vector, okay, to insert a desired DNA sequence into a tDNA region of the TI plasmid. We can insert our gene of interest here, and that is why it is also widely used in plant genetic engineering and also gene transfer in plants. If we want to insert or transfer any particular genes in the plants, we can use these TI plasmids. Suppose we can remove the gene in the tDNA region which is responsible uh, for causing the tumor, okay, the oncogenes, that means the genes which are responsible for causing tumor, those can be removed from the tDNA region. We can cut those region with restriction enzymes. Then we can insert our gene of interest. Suppose our gene of interest is for disease tolerance. I want to insert a disease tolerance gene into a particular plant genome. So what I will do, I will insert that gene of interest which contain the genes responsible for disease resistance. I will insert it to this TI plasmid. Then this TI plasmid will act as a vehicle for transferring that gene of interest into the plant genome. So those plants, they will become disease tolerant. So the development of disease tolerant, stress tolerant plant varieties are achieved through the use of these TI plasmids. But of course, there are also some limitations to the use of TI plasmids. Okay, for example, because sometimes it is difficult to remove the tumor induction property, uh, which this TI plasmid has, and also the absence of unique restriction sites in the tDNA region. So now the next very important vector used in the recombinant DNA technology and genetic engineering is the lambda phage vector. Okay, this lambda phage, the full name is Enterobacteria phage, lambda, or it is also referred to as lambda phage or coluphage lambda. Okay, this is actually a bacterial virus or a bacteriophage, meaning a phage that infects a bacterial species, that is Escherichia coli. So it used to infect the bacteria. This virus is temperate and it may reside within the genome of its host through lysogeny. So we will see how does it resides inside the genome of its host. That means in the genome of the bacteria. This lambda phage is actually discovered by Esther Lederberg in the year 1950, and the wild type of this virus, as I had mentioned, it has a temperate life cycle that allows it to either reside within the genome of its host through lysogeny, or it can enter a lytic phase. Okay, and in the lytic phase it will kill and lyses the cell of the bacteria to produce offspring. We will study in detail its life cycle. This lambda phage, it consists of a virus particle, as you can see here, including a head. Okay, you can see a head containing DNA. The double stranded linear DNA genome is present in the head. Remember, okay, just remember that. And it has a tail. Okay, a tail and the tail fibers. So this is the structure of a lambda phage. Now the phage particle, it how does it infect the bacteria? It will uh, recognize and binds to its host, that is E. coli, causing this DNA in the head of the phage to be ejected through the tail into the cytoplasm of the bacterial cell. Then when this DNA, okay, it has been inserted into the cytoplasm of the bacterial cell, then the DNA, it will circularize, because it is a linear DNA molecule, but it will be circularized at the cos site. We will see what are those cos site. Now, as I had mentioned, it has two types of life cycle: the lytic cycle and the lysogenic cycle. So let's take a look at the life cycle of this virus, okay, when it infects the E. coli cells. Now, just take a look at this figure. First, we will take a look at the lytic cycle. As you can see, the first thing which happened when the phage it attached to the host cell, it will inject its DNA into the cytoplasm of the host cell in figure 1. Okay, you can see that one in red color. It is the DNA of the virus. So this one in red color, the DNA of the virus, as you can see, when it enters into the bacterial cell, the phage DNA circularize. Okay, it will become circularized at the cos site. And you can see besides that there are also chromosome of the bacteria itself. Then what happened when it enters the lytic cycle? This DNA, this phage DNA, it will produce a viral particle, okay, which will lead to viral assembly. So in usually a lytic cycle ensues where the lambda DNA is replicated many times, okay, and the genes for both the head and the tail, okay, as well as the lysis proteins, they are expressed. So we can, this will lead to the assembly of multiple new phage particles, as you can see here in figure 3, new phage particles. But when the more and more new phage particles, also known as the virions, they within the cell, they will lead to subsequent cell lysis in figure 4. When there are so many virions inside the bacterial cell, it will burst, releasing these virions into the environment. Okay, so this is the lytic cycle. But however, under certain favorable condition, as you can see here, okay, when the phage attaches to the host cell and injects its DNA, then the second thing which happen is that the DNA, the phage DNA, it circularized. Okay, what will happen when it enters the lysogenic cycle? Just take a look at this one in red. In figure A, you can see the phage DNA, it will integrate itself into the host cell chromosome, the one in blue and the one in red, you can see. This is because the phage DNA, it integrates itself into the host cell chromosome in the lysogenic pathway. In this state, the lambda DNA is called a prophage. Just remember this important point, that the lambda DNA in this case, in this stage, it is called a prophage, and it stays resident within the host cell genome without any apparent harm to the host cell. Okay, and then the host cell, it can be termed as a lysogen when a prophage is absent. So this DNA right now is referred to as the prophage. Okay, because when the virus DNA is integrated into the DNA, into the genome of the bacteria, at this stage in this figure A, you can see it is termed as a prophage, and the host genome is referred, the host itself is termed as a lysogen. So what happened in figure B, when the host cell divide, these also they will be divide, they will be given to the new offsprings. And you can see in the when the cell division, okay, when the cell division occurs, when the cell divides, these phage also will increase in number. And what happened later on, the phage DNA, it exits the genome by a site directed recombination process. Okay, there is a recombination process whereby this genome of this virus, it gets separated from the genome of the bacteria. Then in this case, it can either again enter, you can see here, it can either become circularized. And from here, just take a look properly at this, uh, just take a closer look at this diagram, girls. You can see here that after D, okay, after this figure D, where this phage DNA, it exits the genome by site directed recombination, then after it gets separated, it will become circularized again like this. And you can see this red arrow, it can enter the lytic cycle. Okay, so after it enters the lysogenic cycle and redivide without causing any harm to the bacterial cell, from there it can again, it might again enter the lytic cycle. So, but in the lytic cycle, as you can see, if it enters the lytic cycle, then this will lead to the production of many virions, and the cell will lyse. Okay, so cell lysis will occur, and the virus will be released into the outside environment. That means they will cause harm to the bacterial cell by killing and lyse the bacterial cell. So these are the two life cycle of this particular phage. So this lambda genome, okay, there's only this DNA, okay, the DNA which is present in the head region of this virus. I'm talking about this lambda genome. This DNA is approximately 49 kb in length. Okay, it is 49 kb in length, but only around 30 kb is required for lytic growth, okay, for the virus to reproduce. As you can see, it has two life cycle. So for the lytic growth of the virus, only 30 kb of this DNA is required. That means 1/3 of the genome of the phage is non-essential. Okay, is non-essential. Even if we remove, that means there will be no harm to the virus if we remove this 1/3 of the genome of the DNA, and it is, we can replace by our foreign DNA of interest. If we have any DNA of interest, we can insert into this lambda genome, and when the virus, it infects the bacteria, it will transfer this virus to the bacteria, and thus it will enter the lytic cycle. Then many virus will reproduce, and in that case we will get many copies of our DNA, our gene of interest. So in that way, this lambda phage, it can use as a vector. Now the lambda viral genome, as I had mentioned, it is about 49 kb in length, or it is actually 48.5 kb, almost 49. Okay, and it is a linear DNA, as I had mentioned, and Here also you can see the small DNA I had shown in this picture at the side, head containing DNA. You can see the DNA molecule. It is actually a linear DNA, but it becomes circularized only inside the bacteria when it enters the host cell. Now this linear DNA, it has a base 12 base, okay, single stranded DNA sticky ends. Just listen to this properly. It has a 12 base single stranded DNA sticky ends at both ends. This DNA, okay, this particular DNA, it has it is double stranded, but it has a single stranded DNA at both the ends. And these ends, they are actually complementary to each other. They are complementary in sequence, meaning suppose if one end of the DNA it has ATGC, okay, then in the other end it should complement, because A it will pairs with T, then T with A, like that. Okay, they are complementary, so that is why they can hybridize to each other. When they hybridize to each other, then only the DNA it will become circularized. Okay, so this region of DNA at both the ends, okay, it is referred to as a cos site. Cos short form for cohesive ends. Okay, so this site it is referred to as cos site. And the lambda genome, it contains an origin of replication, the genes for the head and the tail proteins, and also it contains enzymes for DNA replication to take place. So girls, now I am not talking about the virus, the whole phage. I am just talking about the genome, which is present in the head of this virus. That DNA in the top right corner, you can see DNA which is present in the head of this virus, where it is this DNA which gets transferred into the bacterial cell. So let's take a look at the picture of this. This DNA actually, it is, you can see in this figure in the top right corner, it is a double stranded DNA molecule, and I had just mentioned that at both ends of this DNA it has those sticky ends which can hybridize with each other. Okay, and because they are complementary, so they can hybridize and thus forming a circularized DNA. So in this case, you can see inside the bacteria, when it becomes circularized, it looks like this. Okay, it has genes for making the head of the virus, and the genes for making the tail, and it has the genes for lytic cycle for lysis. It also has the genes for lysogeny, that means when it enters the lysogenic cycle, and it has the origin of replication. Okay, so this is the uh, DNA molecule of this virus, which has become circularized at the cos site. As you can see here, cos for cohesive, okay, cohesive sticky ends. They will stick together, they will hybridize because they have complementary region, and thus forming a circular DNA molecule. So what happened actually here? Now, the genes for lysogeny, just take a closer look at this figure. The one in blue color, this whole portion below, okay, a huge portion near that origin of replication till this other side where I had written here tail, till this portion, all this gene, it is for lysogeny. Okay, so this genes for lysogeny, they are located in the segment between 20 to 38 kb. Now the whole or part of this segment of lysogeny can be deleted, okay, so that we can create a vector, a vehicle to accommodate a large DNA insert. Okay, that means our DNA of interest, we can remove this segment from the lysogeny gene, and we can insert our DNA of interest, and also to ensure that the recombinant phage is always lytic. Why always lytic? Because if you remove this gene for lysogeny, then this DNA, when the virus it infects the bacteria, then it will not have the lysogenic life cycle anymore, because we have removed, right? So we can insert our DNA of interest, and thus only lytic cycle will happen. So when the many virus are produced, then our gene of interest also they will be cloned. They will form, they will have we will get multiple copies of our DNA of interest, which are present inside the head of these newly formed virus. So that means what happened when the lysogeny gene is removed, then that means this bacteria it will not be able to undergo lysogenic cycle anymore. Okay, it will always lytic. So just remember this important point. So let's see how this bacteriophage lambda, the genome, this DNA, how can it be, how we can use this as a vector. Okay, how can we insert our DNA into this? It's simple as in any other vector. You can see this is the first figure here is the genome of a bacteriophage, only that particular DNA. We have to isolate that DNA. Okay, then suppose this I had shown in blue color, this non-essential segment. Why I've written here? Because it is not essential for the life cycle for the virus. It is not essential because we can see, suppose this is the portion responsible for lysogeny. Now this lysogeny it can be removed. It won't cause any harm to the virus. The virus can still live, and it can continue to reproduce through lytic phase. So this non-essential segment can be removed. And if you just take a look at this first figure here, it is a double-stranded DNA molecule, right? And at both ends, it has these what we call the cos sites, 12 bases, cos site, both in the left and on the right. And these are single-stranded DNA. Because they are complementary, so they will join together to form a circularized DNA molecule, but that will happen only inside the bacteria. So it is around 45 kilobase pairs. Now central portion, which is approximately about 15,000 base pair, that means 15 kilobase pair of this lambda genome, is not essential for replication of the phage DNA. So that is why it can be removed. Now suppose you use one of the restriction enzymes, that is BAMH1. I'm just showing an example of only one restriction enzyme. So you cut this portion with a BAMH1 restriction enzyme, then we are left with only the left arm and the right arm with the origin of replication. Remember, because origin of replication is important, so that this DNA, when it's inside the bacteria, it will replicate by itself. And when the central region removed by the restriction enzymes, then we can see this one in orange color. It is a DNA insert, our gene of interest, any gene we want to insert. Okay, so the insertion and also ligation, remember we have to use DNA ligase enzymes for ligation, for sealing these segments, okay, of foreign DNA and the viral DNA. Now after insertion and ligation of this foreign DNA, then the packaging, okay, the packaging of this DNA into the phage we have to do, because at first we have taken out the DNA outside, right? We have isolated this DNA. Then at the last part, after we insert our gene of interest, then we have to insert back this particular DNA molecule into a phage, because then only this phage, when it infect the bacteria, it will transfer the DNA, our DNA of interest, into the bacteria. So this is referred to as a recombinant viral particle. Why recombinant? Because we have recombined our DNA of interest with the DNA, with the genome of the virus. okay, which carry the foreign insert. It is able to infect the bacterial host cell, such as E. coli, and it will replicate to form the leaks. Okay, so the central part of the lambda phage vector, it can be replaced with foreign DNA without affecting the ability to infect cells and to replicate. Remember that if we remove the segment, the non-essential segment, okay, we can remove and replace without foreign DNA, but without affecting the ability of the virus to infect the host cell and also to replicate, because if that happens, then it is of no use. If the virus itself, it cannot infect the E. coli cells anymore, then it will be of no point. So the lambda vectors, they can carry over 24 kb of the clone DNA. Okay, so of the DNA which has been cloned inside the bacteria, and this lambda DNA, okay, you can see here in the last figure, when we insert our gene of interest, okay, this whole lambda DNA, it must be larger than 38 kilobase pair, okay, 38 kb, and it should be smaller than 52 kb in order to be packaged into the phage particles. Remember these small important points which I mentioned, because these they used to come as multiple choice questions in any other exams, okay, competitive exams. So just remember that the size of this DNA, whole DNA, now the new DNA, okay, the lambda DNA with our gene of interest, this orange one, this whole DNA, it should not be more than 52 kb, and it should not be less than 38 kb, so that it can be packaged into the virus again, into the phage particle. So this, we can see, this is the whole process of how we can use this phage particle as a vector.